Plasticity of dendritic spines is correlated with synaptic plasticity and learning and memory. The main purpose of this review is to provide an overview of the recent advances in understanding of molecular mechanisms underlying synaptic plasticity.
Plasticity in the brain is its ability to continuously alter its neural structure, especially by strengthening or creating new synaptic connections. The dendritic spines are the sites of major structural changes in synaptic plasticity. They are tiny postsynaptic protrusions covering the dendrites of most principal neurons in the central nervous system that help to transmit electrical signals to the neuron's cell body. The studies in this review provide insight into the dynamic regulation of biochemical signaling in neuronal compartments during structural plasticity. For example, researchers observed morphological and functional changes in a stimulated spine, but not in its neighbors, indicating long-term potentiation can be induced in an input-specific manner at a single spine. Unique imaging techniques such as fluorescence lifetime imaging microscopy, known as FLIM, combined with 2-photon uncaging of neurotransmitters, have allowed researchers to directly observe the spatiotemporal pattern of biochemical signaling at a single spine resolution.
One of the most important parts of this review is the unique mechanisms that diversify the plasticity in neurons. Dendritic spines have considered to be one of the independent functional units of neurons. That is partly true, but recent studies have also revealed some signaling can spread into the nearby dendrite and spines and even nucleus.
First, synaptic stimulation produces a short calcium transient, largely restricted to the stimulated spine, initiating a biochemical signal transduction crucial for long-term potentiation and long-term depression. When calcium flows into the spine through NMDA receptors, it binds to calmodulin and activates calcium calmodulin-dependent kinase 2 and calcineurin. Induction of long-term potentiation at single spines also activates small GTPases, such as HRAS, Rho, and CDC42. Interestingly, the activation of CDC42 and Ryohei, but not HRAS, is sustained for approximately 30 minutes. Notably, CDC42 activity is restricted to the stimulated spine, whereas HRAS and Ryohei spread over 5 to 10 micrometers of dendrites and invade nearby spines. Activation of small GTPases trigger the activation of two cytoskeleton proteins, cofilin and ARP23, which further regulate structural long-term potentiation via actin remodeling. Spreading signals fine-tune synaptic plasticity in the surrounding area by facilitating and inhibiting subsequent synaptic plasticity. The spreading of signaling proteins, such as HRAS, to much longer distances can activate its downstream effector, ERK, causing long-distance spreading of ERK activity in the nucleus. A future challenge in the study of learning and memory will be to find how the operating principles of signal transduction during synaptic plasticity in vitro can be applied to animals in vivo.
We hope that this review will help other researchers to join forces to studying the spatiotemporal aspects of biochemical computation in neurons, which eventually help us to understand learning and memory.